Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P1 Fund, the Center for Mesoscale Mapping, housed in the Martinos Center. It is our great pleasure today to have Dr. Carol Wilkinson to give us BrainMap talk. Dr. Carol Wilkinson is a physician scientist in the lab of cognitive neuroscience in the Division of Developmental Medicine at Boston Children's Hospital. Dr. Wilkinson earned her MD PhD and completed her pediatric residency at the University of California, San Francisco. There, her graduate work focused on molecular mechanisms of synaptic plasticity. She joined Boston Children's in 2014 as a developmental behavioral pediatric clinical fellow and now continues to see patients in Autism Spectrum Center and Developmental Medicine Center. The Wilkinson's lab research uses EEG to identify biomarker of language development in young children with Fragile X syndrome, Down syndrome, and autism spectrum disorders. In addition, research projects focus on understanding biological and environmental factors that impact early brain development. Ultimately, these research efforts aim to guide the development of effective therapy to improve developmental outcomes and functional independency for children with neurodevelopmental disorders. I would like to remind our audience to address your question at the end of the talk by using the Q&A box or raising your hands. Without further ado, the stage is yours.
Thank you. Great. I'm really excited to be here and to talk to you guys about some of the work that we've been doing. And I'll start off just thanking, so I'm sure I'll run out of time, so I'm going to do this, thanking all of the efforts of the various people that have contributed to the data that I'm going to present today. It's actually 4 different longitudinal studies, so that is a lot of people who are not pictured or named here, but if you can imagine 4 different longitudinal studies over 15 years from largely Chuck Nelson's lab, that is a lot of people, as well as highlighting some of the work of several postdocs in the lab, and of course all of the families and infants. I don't have any disclosures. So my lab's research goal is really to understand the neurobiology of language and cognitive development in children with neurodevelopmental disorders. And to do this, like my bio suggested, we focus in part on children with genetic-based neurodevelopmental disorders, including Down syndrome and Fragile X syndrome, as well as infants that have a sibling with autism, in trying to better understand various factors that lead to the heterogeneity of language and cognitive development. And so with those studies, we have found interesting findings and differences in EEG activity. But in addition to that, we have come to realize that we still really know very, very little about typical development. So we might see something that's different in a 3-year-old with Fragile X compared to a 3-year-old that's typically developing. And then we're like, well, what is that difference all about? And unfortunately, we don't really understand what a typical EEG means for a 3-year-old and what those activity represent. And that is really surprising because we know that the developing human brain is foundational. This is the early period, 0 to 3, sets up future outcomes. As a parent, I and you know how much dramatic change happens over the first year, right? Little babies go from being able to do barely anything on their own to being able to roll, to crawl, to walk. And that happens over that 1.5 period, in addition to dramatic changes in the visual system and the language skills that are built as well. And so we know that infant brain, that 0 to 1 period, the brain is far, far from mature. There are many, many things that are happening within the brain, and understanding that is important. And also understanding that the brain is not an independent system, right? Those changes that happen are a combination of the genetic underlying features of the brain, as well as how the brain responds to environmental stimuli, sensory stimuli, but also impacts that happen during fetal development and onward. And so, as you guys know probably better than I do, the brain, we know from MRI studies that there are big changes that happen during that first year. The brain doubles in size. There's a threefold increase in cortical gray matter, doubling in the surface area, 50% increase in cortical thickness, really all in that first year. And also we know that sensory areas mature first. So, right, and that makes sense. But the synapse number peaks first in auditory and visual sensory areas, and then later in the prefrontal cortex. And similarly, myelination occurs in sensory areas going from posterior to anterior. In addition to that, over the first year, there's also a lot happening in the inhibitory neuron system, right? So inhibitory neurons are generated in the ventral subregion of the ganglionic eminence, and then they have a protracted neurogenesis and migration that is still happening after birth. And so the functional effects of those inhibitory neurons on subsequent cortical maturation and function depends on how well those inhibitory neurons migrate and how well they integrate into the system. In addition to that, those functional effects are also dependent, of course, on the timing of things like when do interneurons switch from an excitatory to an inhibitory neuron. And that also likely happens after birth, although we don't really know that for sure. And then there is also huge amounts of change that's happening in thalamocortical development. So in addition to the inhibitory system, the thalamocortical connections are being made, and those thalamocortical feedback loops and the sort of appropriate stepwise progression that happens with thalamocortical maturation is also essential for cortical brain development. And we know that early insults to fetal brain development in utero infection, in utero drug exposure, hypoxic ischemic injury can all affect thalamocortical connections and therefore also impact cortical development. And so in our lab, we don't do any MRI imaging. We do EEG. And the reason for that is because one reason is because EEG has a really great temporal resolution that allows us to measure spontaneous activity as well as task-related activity. But more importantly, it's very well tolerated by infants, toddlers, and children that have significant behavioral challenges. So for me, some of those genetic disorders that we study are genetic disorders where many of the kids cannot sit still in an MRI scanner. It also allows us to do repeated EEG over the first year to look at those longitudinal changes that may happen. And so for those of you that are not as familiar with EEG, what is EEG measuring? So basically it's measuring the synchronized and unsynchronized firing patterns of mainly pyramidal neurons in the cortex. And of course, the pattern firing of pyramidal neurons depends on many other neurons and the connections that are underlying why the pyramidal neurons fire. And so what we get from this signal, this is what an EEG looks like, right, when we're doing the recording. And one of the ways that we can measure it is with something called a power spectrum, which I'll go over a little bit in more detail. And so when I'm talking about those underlying patterns, that means that with an EEG, we can have somewhat of a glimpse into sensory networks, as well as thalamocortical networks, and as well as the inhibitory neuron system. And so the research question that we have been trying to, one of the research questions that we started to investigate and trying to better understand typical development is how does this EEG power spectrum change over the first 3 years of life? And despite there being a lot of EEG research out there, there hadn't really been longitudinal study of those changes happening early on. And then of course, we want to know how might those changes inform our understanding of neurodevelopment in humans, and how does that also relate to the differences that we see in neurogenetic disorders? And so the punchline, I give you the punchline of sort of what we find, and then go through it in more detail. So the punchline is that we observe transient and nonlinear changes in EEG activity that we think are related to distinct steps in thalamocortical maturation or in inhibitory neuron development, and these have informed some of the findings that we have observed in neurodevelopmental disorders. We are really seeking people's ideas, critiques, collaborations, and thinking about how to further investigate some of our findings. The data that I'm presenting today was recently published, and so you can also read that in more detail. And so the data sets said that there are 4 different studies. 2 of the studies are what we call infant sibling studies. So those are studies where children were recruited because they have a sibling with autism. And so the goal of that original study was to look at predictors of autism. For this, we are not including any kids that have a sibling. We are just using the kids that are in what we call our low likelihood group. So no siblings with autism expected to have typical development. And with those studies, they first were recruited at either 3, 6, or 12 months and had longitudinal EEGs collected as frequently as 3, 6, 9, 12, 18, 24, and 36 months of age. There's another study called the Healthy Baby Study, similarly longitudinal and similar time points, but infants were from our primary care clinic at Boston Children's, which is a largely a more racially diverse, ethnically diverse population. And then the Emotion Project was a sort of combination cross-sectional longitudinal study to try to better understand EEG factors related to anxiety and emotional development. That study recruited kids either at 5, 7, or 12 months of age, and then saw everyone again at 3 years of age. And these all were done in Chuck Nelson's lab, who's head of the labs of cognitive neuroscience, where I did my postdoc, hence why I started looking at the data. And so overall, like I said, this is limited to participants without a family history of autism, and all kids also did not have a later diagnosis of autism. They were removed. And in total, there were 592 infants with over 1,300 EEGs. They are largely a 74% white, 90% non-Hispanic. So not exactly the most diverse population. I think that's a testament to these largely being lab-based studies, and we're working very hard at improving the diversity of our sample. We can talk about that more, but you should know that this is not a very diverse sample. Okay, and so back to this power spectrum. So what a power spectrum basically is that we're taking the electrical activity that we're measuring this is one electrode that we're measuring from the EEG, and we're basically extracting the information to look at the amplitude, the strength of activity that's happening in these different frequency bands. And so this is made up of different types of oscillations that happen either in slow, like delta, which is commonly seen during sleep, or in high frequencies like gamma, which is 30 to 50 hertz. And in addition to these oscillations, there's also underlying sort of noise in the system. And what we get when we look at that breakdown is that we see this power spectrum, which generally has a 1/f curve, with the lower frequency having the highest amount of amplitude and power, and the higher frequencies having lower. This is when we look at all of the data that we've collected, we break it down into different age bins, so color-coded in rainbow format, 2-month-olds in pink, and our 36-month-olds in purple. You can see that there's a large amount of change that happens. We'll go into some of these into more detail. But what I want to also point out is in the EEG literature in the past, most of the time people sort of do analyses that are broken up into these what we call canonical frequency bands. So they'll look at alpha as somewhere between 6 and 9 hertz, or 9 and 13 hertz. They'll average the amount of activity that's happening within that frequency range, and then look at how that relates to some type of behavioral or cognitive or language finding. But what you can see is that there are a lot of shifts that happen, and sometimes that band may not line up with a developmental shift that's happening. And that can cause issues when you're trying to really interpret your findings, and especially the case when we have genetic disorders as well, where they may not align, the differences may not align within where these canonical bands are. And so because of that, over the last, I would say 3 to 5 years, there's been a shift in ways that we look at the power spectrum. And one of those shifts has been to separate out the power spectrum into something we call an aperiodic and a periodic power. And so the aperiodic power is the 1/f fit. So if we think of this as the power spectrum, you can model the underlying curve, and that is thought to reflect non-oscillatory activity. So the underlying noise that's occurring of broadband neural firing, not synchronized. I like, for people who don't think about this as frequently, I like to think about it as your cathode ray tube TV, and you've got the static and it's shh, right? And that is what we're looking at here. There's also growing evidence that the height or the offset is thought to be related to sort of the amount of synapses you have, the amount of neurons you have, but the slope is thought to be related to EI balance, such that the flatter the slope, the higher the E compared to I, and the steeper the slope, the lower the E compared to I. And there's more and more work looking both on computational models, animal models, and human sort of interventions, giving medications and looking at changes in the slope. And then if we remove this 1/f fit, and we subtract it from the spectrum, we then get something that we're calling the periodic power. And that is thought to represent the actual oscillations. It's an indirect measure. We're not doing a time frequency analysis. We're actually measuring the oscillations, but it's sort of a mathematical, just quick and dirty way of saying, okay, anything that has a peak above that 1/f is probably oscillatory. And then we're able to look and say, where is the oscillation happening and what is the strength of that oscillation? You can think of that as turning your channel on your TV and all of a sudden you see colors, right? And so if we look at our data now, a computational package that's used to do these estimates is called FOOF. They just changed their name to SpecParam. I really liked FOOF, but they've decided this is from Bradley Wojtek's group. And so if you FOOF it or if you SpecParam it, this is what our data looks like. So you can see here's the aperiodic activity. You can see that there are some real dramatic changes that happen over the first year of life in that aperiodic component. And then you can more clearly see all of the nonlinear changes that happen in that periodic component. Now, to better describe these, Lisa Yankowicz, a postdoc in the lab, worked on using generalized additive mix models to look at longitudinal changes so that we're not looking at just age bins, but we're looking at the change that happens in these different features. And so I'll start off talking about the aperiodic power changes. And so these are the GAMS models. We've also included sex to see if there are differences between sex. And so what you can see is that in the aperiodic activity, so again, that's the non-oscillatory activity, broadband noise, probably somewhat of a reflection of EI balance. We see that there is generally an increase in aperiodic slope across the first 3 years, and then a dramatic increase in aperiodic offset really for the first year, and then that settles out. When we look at different regions of interest, we see that increase is most prominent in the posterior region. So this is just taking electrodes that are more posterior compared to central, frontal, and temporal. And that maps out with other studies that have looked at synaptogenesis as well as changes in MRI, finding that the visual cortex has the earlier synaptogenesis that peaks before the first year and then declines, and then prefrontal cortex has a slower progression. The other thing to say is that this actually is in stark contrast to the studies that have previously been done when you read about development in these two measures. There's not a huge amount of literature, it's a growing literature, but most other studies have looked in childhood or have started at 3 years of age and looked older. And those studies have all found the opposite, that with age, you get a decreased slope and you get a decreased offset. Some of the hypotheses around that are around pruning that happens, and that would, and when we're talking about the first year of life, we're not expecting pruning, we're expecting large amounts of new neurons being made, integrating, synapses being formed. So I think this makes sense to be different than what previous studies have found when looking at, 3 to adolescence. The other interesting thing to think about with this aperiodic slope would be that this is an increase in slope, which is suggesting that there's a decrease in the EI ratio with age over that first 3 years. And why might that be? One hypothesis that we have is that if interneuron migration is still happening and you're going from excitatory to inhibitory function of those inhibitory neurons, then you might expect to see in that early period a shift from increased E over I to decreased E over I, with increased inhibition coming on board. Take a slight detour to show you what we've seen in our neurodevelopmental disorders. And actually, these studies sort of happened simultaneously. It's not like we saw one and we were looking at it most of the time as the genetic disorders that we were seeing differences. And then as we've been looking at the typical development longitudinal data, it's given us ideas. And so in our Down syndrome and our Fragile X children, these are all from kids that are over 2 years of age. We also see an increase in aperiodic activity, both in Down syndrome and in Fragile X syndrome, with a slight decrease in slope, which would be an increased excitation over inhibition, which is what you would expect certainly in Fragile X, a little more controversial in Down syndrome. And then in our infant sibling studies, so these are infants who are siblings of children with autism, so have a familial likelihood of autism, we see that those infants actually have reduced aperiodic activity at the youngest time point, but they don't show any differences at 12 months. And it's the change that happens longitudinally from 3 to 12 months that is associated with both an autism diagnosis later on, as well as increased language delay. And so this is just sort of demonstrating, right, that if we can understand what is expected to happen over those first years, we can maybe better understand what is different in our neurodevelopmental disorders. Okay, so now looking at some of those periodic power changes, this is showing just the changes that we're seeing in what I would call the theta alpha range, so anywhere from 3 to 12 hertz. It's well described in the literature that there's a shift in the alpha frequency. So in infants, the alpha frequency is thought to be around 4 to 5 hertz, and in adults, it's characteristically 10 hertz. And there's a shift that occurs over those, I would say, from infancy all the way to like 8, 9 years of age. And so we see that similarly here, and you can see it right pretty clearly, right how it shifts. You also see an increase in the strength of the signal as well. So this is all just replicating what previous people had observed. But what was really interesting to us was actually this little pink one. That's our 2 and 3 month olds. And there we saw this double peak that was only really there in the 2 to 3 month olds. This is showing the individual power spectrum, just so you can get a sense of the consistency of kids having both about a 4 hertz peak and a 10 hertz peak. And a reminder that that 10 hertz is really what we usually see in adults, right? So usually it's here, this 10 hertz is even like a higher frequency than what we are seeing on average in our 36 month olds. But this peak goes away. And then we see what the standard shift from 4 to 10 hertz over that period of time. And this is just showing that change of most of the children having 2 peaks at our youngest bin, and that dramatically going away afterwards. So that's one of those transient findings. And so what does the shift in alpha frequency mean? So alpha is very well studied in adults as well as in children. It's associated with visual attention. You turn the lights off, you get increased alpha. If you're focusing on something, you get decreased alpha. And so and it's thought that the thalamus plays a central role in that generation of posterior alpha. But we also have to remember that when we're studying something like this in infancy, it isn't going to be the same as when we're thinking about it in adults, because the circuitry is not yet developed, right? If we're thinking, oh, there's still thalamocortical maturation happening and some of those connections haven't been made, then what we are assuming alpha is or what that frequency means may be different. And one possibility is that before those connections are made, that these shifts in frequency could be related to sort of where in the maturational process we are in the thalamus connecting to the cortex. So the thalamus itself fires both in the theta and the alpha range, and whether it's firing in theta or alpha, or sort of a low alpha, high alpha, is dependent on the inputs it receives. And so sort of understanding that, I think, is something that we might be able to do with EEG data, but also sort of taking hints at this. And someone asked about, these are all awake children. All of the children where they might have fallen asleep are removed and excluded. So these are all awake kids in a parent lap watching a little screensaver video or something like that. And so, right, one of our hypotheses is that the shift from a 5 hertz to a 10 hertz might be reflective of those thalamocortical feedback loops and how those are developing over time. And then this is just a little bit more about I want to show the difference between those two peaks that we see at 22 to 4 months of age. This is just across the different regions of interest. That 2 peak is actually most prominent in the posterior region. And then you can see those the similar plots, but for 4 months. And you see how the proportion of infants with 2 peaks really dramatically goes down. And so what might that be? We're not sure. We'd love to have people's input. One thing that we're curious about is the subplate. And that's because the subplate, from my understanding and my reading after seeing this data and talking to Chin-Fei Chen at Boston Children's Hospital, is that the subplate is this structure that is incredibly important to the development of those thalamocortical connections. And it actually has very mature, it's present in the fetus, likely portions of the subplate are still present after birth, although I think harder to define on MRI. But these subplate neurons are much more mature than the neurons that are in the cortex. And because of that, we think that that connection between the subplate and the thalamus may create a 10 hertz more mature oscillation, because these neurons in the subplate are in a mature state. But then the subplate dissolves and is no longer the major contributor to that cortical to thalamic input. And instead, the thalamus is starting to make connections to the layer 4 neurons in the cortex, which are less mature. And maybe that's why we see that loss of 10 hertz oscillations, and we start to see more of that 4 to 5 hertz oscillation. And as these connections get stronger and these neurons get more and more mature, that we shift to a 10 hertz. No idea if that's actually true. That's just our hypothesis, but we'd love to figure out how we would test that. And I don't think we'll be able to test it with EEG. So I'd be very interested if anyone has ideas on how to figure that out. One way we think we might be able to look at it is actually with our Down syndrome patients, because separate from this, we have EEG from toddlers with Down syndrome. And one of the things that we noticed in that population was that they have a double peak. The vast majority of them had, it's not 100%, it's probably like 50 to 60% of children, they're 13 to 48 months of age, have that double peak. And so here's what the Down syndrome EEG looks like. And then these are a comparison with our typically developing 2 to 4 month olds. Okay, so that's a very big age gap, but they look quite similar. And this is comparing Down syndrome in that orange color with age-matched and developmentally matched comparison groups. And this is showing you on the individual level, again, that consistency. And so maybe there are clues that we can look at in Down syndrome, maybe in children, MRIs from children with Down syndrome, or EEGs, or other ways of, or animal models of trying to sort of see whether there are remnants of the subplate that are still there, or just sort of differences in development in that area that would lead to this double peak. Okay, and so then we also had all of these changes in the beta peak, and actually it was this beta, this section here that got me most interested when I first graphed out all of the changes that we were seeing in the power spectrum. And that was because there was this, what I'm calling a trough. And you don't really think about troughs in power spectrum. And so it was just a little bit hard to wrap. I still don't really know how to interpret it, but it was clearly there. And that over time, over age, a low beta peak emerges. So you can see it starts to emerge around 9 and 12 months of age, and is pretty consistently there in most EEGs by 36 months of age. And we also see major changes in this high beta peak. So here's the low beta amplitude that increases, and you can see these are the proportion of children that have a low beta peak going from our youngest to our oldest kids. And then in the high beta peak, what you can see is that this, I'm also going to point out that this high beta peak is at 30 hertz. And going back to what most people will do, and in the past, many people have looked at beta and they will look at 12 to 30 hertz, or they'll look at gamma 30 to 40 hertz, 30 to 50 hertz. But if you did that in children that are between 4 and 4 months to 36 months, you're cutting right in half of that peak, right? And actually many studies that we had looked at actually excluded, sort of filter out everything above 30 because they thought that this was noise. But it's clearly not noise because there's a developmental pattern. It increases, it decreases, and then it shifts to a low beta peak. So, and this was very consistent across individuals. It's not just sort of like the mean. And so what do these beta things, we still don't know, but we're loving to have more feedback. But adults sort of beta, again, that 13 to 30 hertz range is associated with sensory motor processing. As you're making or planning movements, you shift the amount of beta activity that you have. It's also thought to be important for working memory and sort of visual perception, combining different pieces of sensory information. We also were thinking a lot about what does the emergence of a peak at that low frequency around 15, 20 hertz, what does that mean? And does it reflect some type of other maturational process? And again, like alpha, low beta is also thought to be dependent on thalamocortical maturation. The LGN also produces beta oscillations. And the other interesting thing is that this emergence that we see in the low beta peak starting around 10 months of age coincides with the time that infants also start having alpha coherence in association with anesthesia. You might be asking, why on earth did we think about that? Well, that's because I had a K23, and Patrick Purdon was my mentor, one of my mentors for my K23, and he's an anesthesiologist and had been looking at alpha coherence, and had Laura Corneliuson was at Boston Children's and I knew about her work looking at longitudinal infant data, but under anesthesia. And so prior to 10 months of age, anesthesia leads to an increase in alpha power, but not coherence. So coherence is when those rhythms are happening like timed together across both sides of the frontal cortex. And so prior to 10 months, babies will undergo anesthesia and have increased alpha activity, but it's not coherent across the two cortices. After 10 months, infants start to, not all of them, have stronger and stronger alpha coherence, and it's thought that that is reflective of a step of thalamocortical maturation, that the response to the anesthesia is linked to that connection. And so we said, oh, well, the timing here is really interesting. If we make the hypothesis that infants that have a low beta peak also or have a mature thalamocortical connection, then we would hypothesize that those infants with a low beta peak would have stronger alpha coherence when they were under anesthesia. And it turned out that one of Patrick's other K mentors, Jerry Chow at Montefiore, he was collecting EEG in infants, both collecting baseline data and data under anesthesia. And so we were able to look at a very small number of infants, 25 infants that were in this sweet spot of an age range between 9 and 14 months, who may or may not have a low beta peak, who may or may not have strong alpha coherence. We were able to test whether we would see that association. And we did. And so here we see that the infants without a low beta peak had lower alpha coherence than the infants that did have a low beta peak. And as we expected, it's about half and half that did or did not have a low beta peak. So super preliminary. He's collecting more and more data, and so we'll be able to look at this with a larger data set. But sort of one interesting way of trying to dissect out the circuitry using EEG and something like anesthesia, sort of to test out the manipulation of a circuit that is developing over time. And so then what about the high beta peaks, this 30 hertz peak that we see increase and decrease? One of the interesting things about this peak is that it is also seen in some neurogenetic disorders. One that is well known is DUP15Q, which is associated with autism. It's a duplication in GABA receptive subunits. There's known to be GABA dysfunction. And they consistently have a peak, strong beta peak at 23 hertz. So not quite the same frequency that we are seeing, but sort of in that same neck of the woods. And we also know that in many kids that are receiving GABAergic medications for like seizures, they also have an increased peak around 30 hertz. So it's possible that this is somehow related to the GABAergic system and how that's changing over development. We also see that same 30 hertz peak in our Fragile X patients. So these are 3 to 7-year-olds with Fragile X. And so very, very different age range, right? This is a 3-year-old here in purple, no prominent beta peak. These are the age matched and cognitive matched groups. No gigantic 30 hertz beta peak, but here in our Fragile X group, we see that, suggesting that maybe there's some type of delay or altered maturation that's happening. And then also interesting for Fragile X, which is a little bit different than what happens in DUP15, which is that this peak does still seem to be associated with age. So the darker lines are older kids with Fragile X, and the pink lines are those that are younger. And you can see there is a shift still that happens with age. It's just happening a lot later. And there has been some work looking at whether there are delays in the switch from excitation to inhibition, the GABA polarity switch in Fragile X. And there's some mouse data that suggests that it is delayed. And so maybe that is what we're seeing in the timing maybe of when those things are happening, or reflective of when we see that in typical development of the beta peak emerging and going away. And maybe what we see in Fragile X is sort of remnants of some amount of GABA not making that excitatory to inhibitory switch. So in summary, So what we're seeing are EEG features that change dramatically and non-linearly over the first 3 years. We see changes that are both related to the amplitude, but also the peak frequency. And that confirms that really the classic sort of canonical band analysis, especially when you're looking early on in infant and toddler development, is not optimal and also not a great way of looking at genetic disorders. And then we also are seeing these changes in alpha and beta that we think reflect the inhibitory system, maturation of the inhibitory system or thalamocortical connections. That includes that double peak that we see, the low beta peak and the high beta peak. And so what are some of our future directions? So one is can we, if we think that these EEG measures are so associated with age, then we should be able to predict what age a child is based off of those EEG features. That is work that Linko has been working on, and I'll detour and show you our findings, which is that with the sort of overlapping data set, we're able to predict age to about 90 days accuracy. We've tested out multiple different models, and there's somewhat of a range of how well they are predictive. And then what we ultimate goal, one of the goals clinically for this would be to use this type of analysis to be able to identify children that may have some type of developmental delay, with the assumption being that if your brain is far, when we estimate your age, it's far from what your chronological age is. That means that there are some atypicality in how your brain is developing. And so we've tested that by looking at the accuracy of the prediction of children that have autism based off of our model, as well as children that have Down syndrome. And there is, when we sort of subtract what their chronological age is and their age estimate by the model, and in autism, we see that there are differences, but not until really that latest time point, the 36-month time point, whereas in Down syndrome, we see pretty striking differences in the age prediction throughout throughout their age. And that kind of makes sense because there's a lot of heterogeneity in autism. Not all children have huge numbers of developmental delays, where in Down syndrome, it's very consistent that children will have motor, language, and cognitive delays. And then, of course, we also want to be looking at the trajectories of neurodevelopmental disorders and seeing sort of where do these changes happen. Do the Fragile X infants at 3 months of age, do they still have a 30 hertz peak, or do the Down syndrome kids ever lose their 2 peaks, right? And sort of understanding that process. We're also trying to better understand these peaks in the alpha range using phase lag coherence analyses to try to characterize those better. And then we're collaborating with Jerry Chow to also recruit kids with Down syndrome to his anesthesia analyses as well. Yeah, and I will stop there and take questions.